Let me proceed with the first event of the day, third plenary session, Biochemical Signaling in Metastasis. I kindly invite Dr. Dehan Gunasekara, Consultant Clinical Oncologist, and Dr. Jalia Jayasekara, Consultant Clinical Oncologist, to share the session. Over to you.
Good morning, everyone. First plenary for the day is Biochemical Signaling in Metastasis. Speaker is Dr. Hasini Jayatilaka. She is an acclaimed cancer researcher, scientist, TED speaker. She grew up in Colombo, Sri Lanka, and pursued her BS and PhD in Chemical and Biomolecular Engineering at Johns Hopkins University. At 28, Hasini made it the 2018 Forbes 30 under the science category for her extraordinary research work. She discovered a novel synergetic signaling pathway between interleukin 6 and 8 that is responsible for the regulating metastasis. And she is here as the first speaker today. Over to you, Dr. Jayatilaka.
Off with me studying cancer metastases. Metastases, as you may or may not know, is the spread of cancer from a primary site to a distal site through the circulatory or the lymphatic system. 90% of cancers are because of the process of metastases. For instance, a female patient with breast cancer doesn't succumb to the disease simply because she has a mass on her breast. She succumbs to a disease because it spreads from the primary site, which is the breast, to the lungs, the liver, or the lymph nodes, or the brain, where it becomes unresectable or untreatable. So metastases has two main drivers, which is tumor cell migration and tumor cell proliferation. My initial work focused on tumor cell migration, where I studied cells in how they move in a two-dimensional collagen 1 coated plastic dish, as well as a three-dimensional collagen 1 matrix. And as you can see from these images, the exact same cell, which is an HT1080 wild type cell, shows different phenotypes in the 2D setting versus the 3D setting. Now, most conventional studies have been done on 2D flat plastic dishes, which really don't recapitulate the conditions that the cells are exposed to in our bodies. In our bodies, they're exposed to the extracellular matrix, which is predominantly made up of collagen. And so you can see that in 3D that the cells are more elongated and they have these arms called protrusions. And so these protrusions can hold on to the collagen 1 fibrils and pull themselves through the matrix. And so, as an undergraduate student, I was tasked with this very difficult task of counting up to what was 6. And so I had to quantify and also qualify these protrusions that were coming out of the cell body. And so I coined the protrusion coming out of the cell body zero generation, and those coming out of the zero generation first generation, and those coming out from the first generation second generation, as shown by this cartoon. And what was interesting was that the second generation protrusions were less prevalent than the zeroth and the first. So it was extremely surprising to me to see that at a particular time point in the same frame that we were seeing second generation protrusions being produced. And so that kind of indicated that there was some sort of coordination between the cells. And so I dove into the literature to really figure out what was happening, and I really couldn't find anything at that point in time. And so the closest thing I was able to find was in bacterial cells, not in cancer cells, where bacterial cells would perform this action called quorum sensing, where they can sense and produce signaling molecules that allow the whole population to initiate a concerted action. And the cause or the driver of this concerted action was a chemical signal between the bacterial cells. And so based on that, I formed this very ambitious hypothesis that said, as cancer cells proliferate and increase local cell density, secretomic profiles change, leading to an increase in cell migration. And so I tested this hypothesis by simply placing a known amount of cells in a three-dimensional collagen 1 matrix and let them grow and move around for a period of 5 days. And what I found was that as the cells proliferated over the period of 5 days and increased their local cell density, that you could also see an increase in cell speed. Now, I wanted to find out if this was in fact an artifact of cell density, or this was because we were seeing all these good stuff, soluble factors sort of accumulating in the collagen 1 matrices and causing an increase in cell speed. And so to determine the effect of local cell density, we measured the distance between cells at different cell densities and sort of correlated the distance between the cells to particular cell densities. We then placed those cell densities in fresh collagen 1 matrices, and for this experiment, we use HD1080 wild type cells, which are a type of fibrosarcoma or bone cancer cell line, which are commonly used to study cell motility. And what we found was this cell density dependent migration, where as the cell density increased, you could see that the cell speed increased significantly and then would plateau off. When we looked at other motility parameters such as persistence distance and invasive distance, again, we saw a similar trend indicating to us that cell density dependent migration was in fact present in this particular cancer cell line. We then wanted to figure out, was this an artifact of just the fibrosarcoma cells, or could we see it in other types of cancerous cells as well? And so we used multiple different cell lines. Some of those are the MDA-MB231, which is a breast cancer cell line. Again, we saw a very similar trend to what we had seen with the HD1080 wild types. We also saw cell density dependent migration in the U87, which is a glioblastoma cell line or a brain cancer cell line. However, we did not see the cell density dependent migration in MCF7s, which are tumorigenic non-metastatic cells, and WI38s and MCF10As, which are considered normal cells. And so what we found was that cell density dependent migration is unique to tumorigenic metastatic cells. And so we also conducted a bunch of different experiments to really look at if this effect that we were seeing of cell density dependent migration was a result of a physical cue or a biochemical cue. And after running multiple experiments with our collaborators at Yale University, what we found was that it was indeed a biochemical cue, which was interleukin 6 and interleukin 8. Now, interleukin 6 and interleukin 8 have been shown on their own to have an effect on cancerous cells. They've been shown to play a part in angiogenesis and sort of inflammation in cancer, but all this was sort of shown alone. And so here we were showing that interleukin 6 and interleukin 8 had this synergistic mechanism which was able to enhance cancer cell migration and thereby enhance metastases. So knowing that interleukin 6 and interleukin 8 were responsible for cell density dependent migration, we hypothesized that by blocking this interleukin 6 and interleukin 8 from binding to their cognate receptors, that we could block cell density dependent migration and essentially slow down the rate of metastases to the different organs in the body. And so to test this, we conducted experiments with preclinical mouse models where we injected MDA-MB231, the breast cancer cell lines, into the mouse memory fat pad. We then treated the mice with either saline, which was the control group, tocilizumab, which is an IL-6 receptor blocker, reparexin, which is an IL-8 receptor blocker, or a combination of tocilizumab and reparexin. What we found during the weeks of conducting this experiment was that the tumor growth, the size of the tumor really wasn't affected by any of these treatments. However, at the end of the experiment, when we measured metastases to the lungs, liver, and the lymph nodes, what we found was that the treatment groups had significantly less Mets than those in the control group. As you can see from the pictures of the lungs, you can see that there are less Mets in the treated groups versus the control group. And you can see that the combination therapeutic of tocilizumab and reparexin had the most effect where you say less metastases to the lymph nodes. And so here, in this particular process, we were able to map out the signaling mechanism using a number of different techniques, including RNA-seq, where we showed that interleukin-6 and interleukin-8 can bind to their cognate receptors, activate the pathway of JAK2 and STAT3, which then activates WOS3, which in return increases the number of protrusions and then increases cell migration. And so when we block these cognate receptors of interleukin 6 and interleukin 8, the cells become less invasive and we are able to significantly reduce metastases to the lungs, liver, and the lymph nodes. So at the end of the experiment, we did publish in Nature Communications, which was extremely exciting. But the question came back to, cool, so what? Which is true because if I was a patient that was given this drug cocktail of tocilizumab and reparexin, and my doctor said I would be fine, which may be true because I wasn't having any metastases, but my tumor kept growing, I think I would be very upset. And so we went back to the drawing board, really thinking about how can we target both tumor growth and metastases to improve outcomes for patients. And so we went back to our collaborators at Yale University, where we conducted another high throughput secretomic profiling assay. And this was conducted for longer periods of time. And what we found was that in addition to interleukin 6 and interleukin 8 showing up in relatively high concentrations and showing a really good relationship with cell density, we also saw the effect of EGF, epidermal growth factor, having a relationship, an inverse relationship with cell density. We then went and looked at patient data where you can see that patients with high IL-6, high IL-8, and low EGF have poorer outcomes than those with low IL-6, low IL-8, and high EGF. And so we hypothesized by blocking the EGFR, which is the receptor for epidermal growth factor EGF, in addition to blocking the cognate receptors of IL-6 and IL-8, we can essentially block cell migration and cell proliferation, which would lead to reduced tumor growth and less metastases. And so we first tested it out in vitro, where we did a combination therapeutic of tocilizumab, reparexin, or cetuximab, which we call TRC. And as you can see, the combination therapeutic of TRC had a significant effect in reducing proliferation in this breast cancer cell line, as well as the fibrosarcoma cell line. And it also had a significant effect in decreasing cell migration at a low density as well as a high density. And so again, we conducted preclinical mouse models. And here, in addition to the MDA MB231 mouse model that we conducted previously, we also conducted a model using a patient-derived xenograft, where you take a piece of tumor from a patient and you can actually implant it in a mouse and see what the response is to certain treatments. And here you can see that as shown previously, the tumor growth with the tocilizumab and the reparexin really had no effect on sort of the profile of tumor growth. However, when you treat the mice with the combination therapeutic of TRC, you can see that tumor growth is significantly decreased. There's also a significant decrease to metastases to the lungs and the liver, as well as the lymph nodes. And again, here in the PDX model, the patient-derived xenograft model, you can also see significant decrease to the tumor size, as well as metastases to the lungs, liver, and the lymph nodes. So we currently have many ongoing projects based on some of the work that I did as a grad student, and part of it is really thinking about how can we develop better combination therapeutics that really target both tumor growth and metastases. And here for pancreatic ductal adenocarcinoma, which is a highly metastatic cancer, we are currently testing the combination of tocilizumab, reparexin, and gemcitabine. And as you can see, the triple combination of TRG has a significant effect not only on tumor growth, but also metastases as well. And here I think it's important to note that gemcitabine is a commonly used chemotherapeutic that's used to treat patients with PDAC, and showing that the addition of the T plus R combination, which is the tocilizumab and reparexin, has a significant effect on reducing tumor growth and proliferation. One of the other questions we also asked was, having given patients multiple lines of therapy is not optimal. And so we asked the question of, can we actually develop our own molecule that will sort of do the function of the tocilizumab and reparexin? So limitations of the combination therapy include the small molecule, which is the reparexin, that lacks specificity, and as well as, dosing complications. You don't want sort of many side effects from the individual therapies. And so we decided to design and develop a bispecific antibody. And these are the two formats that we've developed so far, which really enhances affinity, increases specificity. It also reduces drug resistance, and then also facilitates therapeutic translation. This is true for a fact because we just formed a startup company around it, and now are planning to go into human clinical trials for this new type of medication. And so preliminary data shows that this particular bispecific antibody, the two formats, perform so much better than the tocilizumab and the reparexin in combination. And we see this here in our fibrosarcoma cell line, the HT1080 wild type cell line, as well as in the breast cancer cell line, where both bispecifics are performing so much more better than the combination therapy of tocilizumab and reparexin. And we are super excited about this work, and the fact that we can translate this into the clinic very, very soon. So following my stint at Johns Hopkins University, where I got my PhD from, I decided to continue studying understudied diseases, continuing to study processes such as metastases and other sort of mechanisms that are responsible for relapse. And so I pursued a postdoc at Stanford University in the Department of Pediatrics, where I mostly studied neuroblastoma. So neuroblastoma is the most common extracranial tumor in children and accounts for about 50% of all cancers in infants. They really don't have very good treatment options. Treatment options currently include aggressive chemotherapy, followed by resection, followed by high-dose chemotherapy with stem cell rescue, and just the list goes on and on. And approximately half of the children who are treated for high-risk neuroblastoma and achieve an initial remission will eventually have the disease come back. And those who do survive this neuroblastoma disease. They have severe deformities due to the chemotherapeutics, which can include sort of deformities in the heart, kidney function, as well as some of these patients are infertile. And so there seems to be a huge unmet need in neuroblastoma. And so that was one of the reasons I decided to pursue doing work in that particular field. And so part of my work included looking at synaptophysin, which is a synaptic vesicle glycoprotein that is present in neuroendocrine cells. Interestingly, synaptophysin is used as a marker when children are diagnosed with neuroblastoma. It's sort of looked at, and check mark is used whether to say it's present or absent, but nobody really knows what its function is. And some work done by other groups have found that variable synaptophysin expression has been shown to be found in circulating tumor cells. So there is some indication that synaptophysin could play a role in sort of relapse and metastases. And so, with that in mind, I started off looking at patient outcomes. And interestingly, what I found was that those children with neuroblastoma who have a high expression of synaptophysin had better outcomes than those with low expression of synaptophysin, which was extremely interesting. And so a quick, simple study in vitro where we knocked down synaptophysin in multiple neuroblastoma cells showed that when neuroblastoma is in low quantities or even missing in some cells, that they have this elongated, almost mesenchymal phenotype, which indicates that they are more invasive or more metastatic. And so by sort of doing multiple really cool, really fun experiments, we were able to show that synaptophysin, low synaptophysin, increases EMT, which is the epithelial to mesenchymal transition, which increases cell motility, which in turn increases neuroblastoma metastases. And so we were able to map out the pathway to show that this protein called TREX were involved in synaptophysin-mediated metastases, and that by blocking it using a TREK inhibitor called LOXO, we could essentially block synaptophysin-mediated metastases. And so we also hypothesized that, a combination of LOXO with a standard of care therapeutic, such as vincristine, which is a chemotherapeutic, could target both tumor growth and metastases and have better outcomes for children diagnosed with neuroblastoma. And so here, again, we conducted preclinical animal models where we treated the patients with LOXO as well as LOXO in combination with vincristine, which we coined LV, and as well as vincristine, where here we call it Vin. And you can see that those mice that were treated with the combination of LOXO and vincristine showed better outcomes where there was less tumor growth as well as less metastases. And here we measured metastases to the bone marrow because that is the most common site of metastases in children.
Thank you, Dr. Hasini Jayatilaka, for that excellent presentation. We clinicians appreciate the great work that researchers like you do. Without your help, we would not get anywhere. And we appreciate the great effort that you have taken in both Stanford and Yale. Hasini, just before we get on to the questions, we'd just like to mention that the Sri Lanka College of Oncologists has a cancer research unit, and we would like to collaborate with you on your work, and that will be helpful for both the Sri Lankans as well as we could have more communication with you over this. Following your excellent presentation, there have been many questions. You have answered some of the questions, but that is only to the person who has questioned you. So can we make it public, and can I ask each question, and could you reply in the same way you have done over TV? The first question, Hasini, is what are the cancers highest in the cell density proliferation?
Hello, doctors. Thank you so much for having me and inviting me for this talk. I am so thrilled to be here today, and thank you for the invitation to collaborate with your research unit. I would love to collaborate and love to connect you and any other researchers with sort of my resources here, and also see if we can do some really good collaborations through my startup company, which is Atlas Exomics. I'm super happy to answer all the questions that have come in, and they've been absolutely excellent. So thank you to all who sent them in. So the first question was, what are the highest cancers with the highest cell density proliferation? This is predominantly aggressive cancers, and I was particularly interested in breast cancer just because of what high incidence there are and sort of the rates of relapse as well. Here in the US in particular, there is a push to study breast cancer. There's a lot of funding, and so that was one of the reasons I went down that particular path. But in all honesty, I'm sort of very interested in aggressive cancers, understudied cancers, and those include pancreatic cancer, non-small cell lung cancer, and ovarian cancer in particular as well.
Thanks, Hasini. The second question is the tendency to metastasis with increased cell density related to tumor hypoxia?
That is a great question. So we actually thought that it might be related, and we actually initiated a project that led me to really look at HIF1 and HIF2 in the context of cell density dependent migration. And what we found was those particular markers of hypoxia weren't upregulated when we saw cell density dependent migration. So we don't think this is an artifact of hypoxia, but I think there is a situation in which if the HIF1 alpha sort of, or the HIF2 pathways are upregulated, and then we're also seeing sort of this upregulation of the cell density dependent pathway, that it could be sort of an enhanced effect that we're seeing, and that could lead to more aggressive tumors.
Thank you again. There's another question. How does your work relate to metastasis dominancy? Where is cancer relapse? Why is cancer relapse several years after staying dormant?
That's a great question, and that's something that we are currently exploring. And some of my mentors and some of my friends who are working at University of Southern California at the Lawrence Ellison Institute are in particular very interested in that. And now we've collected the appropriate samples that we're able to sort of now retrospectively look at what are sort of the particular pathways that are upregulated in these cancers that relapse after many years. Is sort of the IL-6 and the IL-8 pathway upregulated? We are kind of seeing hints of that right now, but we're still studying and still trying to understand it.
Thank you. There's one more question. In humans, is there a high incidence of allergic reaction as a barrier to the use of multiple monoclonal antibodies practically?
Yes, I think you see a lot of cases, and this is something that I saw while working on neuroblastoma and even acute lymphoblastic leukemia in children, where there were a ton of side effects of not only using monoclonal antibodies, but also CAR T therapies. And this is, I think, predominantly due to off-target effects. And this is why it's really important for us to develop bispecific or even trispecific antibodies or CAR T therapies that can directly target the cancer and limit off-target effects.
Thank you. There's another question. Why do you think some human tumors preferentially have secondaries in lung than in liver, bone or brain? For example, sarcomas to the lung.
Yeah, I think so. That's something that we're still trying to figure out. But I suspect that it's because there's a whole lot of cytokines in the lungs that's causing sort of this chemotactic, sort of attractive environment to the cells. We know that in particularly breast cancer, the first site of metastases, or the most common site of metastases, is the lymph nodes, and then you see it more in the lung. I just think that the lung is sort of a really good place where they have sort of all the nutrients and the oxygen and like all the other cytokines and chemokines that are necessary for these particular cells to flourish and grow and cause the most amount of damage.
Thank you. Another practical question. Are IL-6 and IL-8 inhibitors currently available in practice?
IL-6 inhibitors are available. So tocilizumab is currently used to treat rheumatoid arthritis. And I believe it's also being used in the treatment of COVID-19 in some cases, particularly when it comes to the cytokine storm. We are seeing the development of IL-8 antibodies that are currently in clinical trials, but to my knowledge, there aren't any that are currently used in sort of other diseases at this point in time.
Thank you. There's another question. Synaptophysin blockers will help in prevention of metastasis only or tumor reduction as well?
We believe both. So the data shows that synaptophysin-mediated metastases is sort of one of the factors in neuroblastoma. And so by blocking it, you can see that there's a reduction in metastases to the bone marrow as well as to the liver. But I think by blocking it, you can also sort of reduce some of the side effects of cell proliferation as well. So I think once used in a combination therapeutic, it can have sort of this dual effect of targeting both metastases and cell proliferation as well.
Thank you. One of our researchers, Nuraj Joseph, from our Sri Lanka College of Oncology Research Group, wants to know why fibrosarcoma was chosen for the investigation in your study.
Yes, so HT1080 fibrosarcoma cells are the most commonly used cells in cell motility. This is because they're sort of easy to study. They've been easy to study in the two-dimensional setting. They kind of move really well. And so that's sort of the common choice used by most investigators. But as we're sort of studying those mechanisms and understanding it, we're moving into other particular types of cancers as well. In my talk, I talked about, not only using breast cancer cells, but we use glioblastoma as well. A lot of the projects that I've been working on following that also have included PDAC as well as ovarian cancer cells as well. So you can do these migration assays with other types of cells, but sometimes it's just easier to start off with the fibrosarcoma cells.
Thank you, Hasini. We have many more questions. We'll go on. What are the impact on collagen function by these suggested treatments for breast cancers, as we may have to use specifically in elderly populations, especially in
so collagen has a really interesting relationship with cell migration and even proliferation to some extent. Work that has been done from my colleagues have shown that, as you increase the collagen concentration, that the cells may get more aggressive in terms of motility. And that's really interesting because we know that as you sort of age, you see more stiffening, or like even in the sense of having breast cancer, you see this stiffening in the collagen fibrils, which actually is now showing to have an impact on cancer cell migration as well. So I think sort of really studying that and understanding that. And yes, in elderly patients, it's not only the impact of collagen, but it's also like other factors of aging, which another colleague of mine has showed that it can be a factor of the cells just being more motile or less motile, or it can also be the cytokine and the secreted proteins, or any of the other signaling proteins around the cells having an impact on them.
Hasini, in neuroblastoma, anti-CD3 antibodies also play a role in addressing metastatic disease. Is synaptophysin pathway inhibition, does it play a synergistic role in this section?
That's actually a great question. That's something that we have not yet really explored. Anti-GD2, I think, is showing a great hope in neuroblastoma, but there are still sort of where it's failing, and this is in particular of the very high-grade neuroblastoma that we're seeing in some patients. And again, those patients have sort of very aggressive chemotherapeutic options only available to them. I think the anti-GD2 is a great sort of starting point, but there's still a lot of places where it kind of falls short. And I think that's where combination therapeutics can really come into play. And maybe that's a combination therapeutic of anti-GD2 plus a couple of other chemotherapeutics, or maybe even adding a corticosteroid would make it more impactful.
Thank you, Hasini. Are IL-6 and IL-8 blockers effective in already metastatic tumors as well, or is it more in the setting of preventing metastasis in non-metastatic setting?
So we believe it's both. So our first study, which I talked about, is showing preventing metastases, but we've also done studies, and this is work that's continued on in my mentor's lab, Dr. Denny Woods at Johns Hopkins, where he's shown that this is effective in mets that have already formed as well. So I think the point and what we're hoping, especially in our clinical trial, is to show that yes, it's good at preventing metastases and also shrinking some of the mets that are also present, but it's also really effective at causing remission. The whole point of thinking of our therapeutic is to really give sort of a life where the patient is sort of in constant remission, which they can go on to live a better or continue to live their lives, and maybe some other disease will sort of affect their well-being. But in terms of our strategy is to prevent future metastases, but also keep the disease in sort of a constant state, so that moving forward it's more easily manageable by not only the patients, but also caregivers and also their physicians as well.
One last question. In a CUP syndrome, why metastatic site prominent while the primary tumor remains undetectable?
That's that's also a great question, and I don't personally have the answers to that. That's not something that I've worked on, but I hope that there's more research into that and we can gain more insights into that in particular.
Hasini, it was great listening to your talk, and all your answers were very, very supportive of our thinking. And on behalf of the Sri Lanka College of Oncologists, we thank you very much for taking part in our college sessions. And we will definitely get in touch with you with our cancer research group. Thank you very much.
Thank you very much.